speaker of session 1 is Associate Professor Anai Gonzalez-Cordero, and I feel very privileged to be able to introduce her today. She is the reason that we're actually all able to be here today. She's the one who has been successful in the Medical Research Future Fund for stem cell therapy and has allocated a part of that grant to be able to put on these events for families because she understands the value and the importance of doing that. So big thank you to Anai. So Anai is a Val Rosenstrauss Fellow. She's a group leader of the Stem Cell and Organoid Facility at the Children's Medical Research Institute, or CMRI as you may hear, and she's also an associate professor at the University of Sydney. She is a leader in the field of stem cells and using them to develop novel therapies for inherited retinal diseases. And she actually began her scientific career in the UK, and so we're very lucky that she's relocated to Australia to continue her research program, which she will be sharing insights to today. So please welcome to the stage Associate Professor Anai Gonzalez-Cordero.
Thank you, Eden. Thank you everyone for being here, really. It's almost a dream come true because we did a lot of these days in the UK and they were so amazing and valuable for us. And I always wanted, when I relocated 5 years ago, I really wanted to make this happen here. We have Safe Site Days every year, but you know, to be able to share the research that we do and have the discussions that, you know, perhaps we're not quite there yet with treatments, but there is some hope. And so much has happened in the last 15 years in research, and being able to share that with you, it's great. And I hope you know, mingle, interact, go to the posters, buy raffles. We're actually going to have a demonstration here on DNA extraction if you want to have a look at that later on. Jennifer is going to be doing that. So just enjoy the day. My job here is to introduce stem cells, which are my second favorite cells. I have to say that photoreceptor cells, the light sensing cells of the eye, are my favorite cells, but stem cells are pretty special. So I'm going to introduce stem cells, what we can do with them in the lab. I don't have time to go through over all the projects that we do and all the amazing research, but again, go to posters and interact with the researchers that are here today to talk to you. So this is the team, and they are the ones in the blue shirt around here. So we are studying inherited retinal degenerations to try and develop new therapies. And to do that, we use stem cells. So our bodies are made of 30 to 40 trillion cells, right? So we are very complex individuals, lots of cells forming the body. And all these cells are formed from the stem cells. So very early on in the embryo, we have a region that is capable of giving rise to all the different cell types in the body. So your heart, your eyes, your brain, and so on. So what we do, and what we used to do in the lab, was to actually isolate the cells from this very early embryo and be able to put that in the dish in the lab and grow these cells. And here actually I'm showing an image down the microscope of what these cells look like together as a colony. They like to grow as a group. And as Lauren explained, they can self-renew, so they just replicate themselves indefinitely. And they're so special because they are in this state that we call undifferentiated state. So they, at this point, they're basically just sitting there waiting to get instructions on what they will become. So they are undifferentiated. So they're that basic cell type that forms everything. And what we can do in the lab is grow these cells, because they like to proliferate a lot, bank them, and then use these as cells to study diseases. And how do we study diseases? So Lauren touched on this as well. In 2012, Shinya Yamanaka won the Nobel Prize for this amazing discovery, which was getting a cell from your skin or a cell from your blood and reprogram, so bring the cells back in time into a stem cell. So now you have an adult cell that is simply your blood, and you can give 4 different factors, so it's 4 different proteins inside in the lab. We get your blood, we grow this blood in the lab. We basically add 4 different proteins in there, and these cells now can go all the way back to being a stem cell. Very similar to the stem cell that we got from the embryo in the past. So now, first, we don't need to use embryos anymore, and I can generate stem cells from any patients, any healthy individuals in the lab. And we can grow these cells indefinitely, and we can market these cells. So what it means is that we have these cells growing in the lab that have the same genetic material because they came from a patient with RP, a patient with LCA. The same disease, same mutation that is there in the eye, it's also present in the stem cells. And when we grow these cells, we can now study. But of course, if I study a stem cell is very different from an eye cell, right? So I need to have a way of making the stem cells now to go all the way back to adulthood and become the cell that I want. In this case, retinal cells, a mini retina that we can grow in the eye and study the conditions. So that's what we do, and that's what we know how to do in my lab. We know how to induce the stem cell to become a photoreceptor cell, to become a ganglion cell, to become any of the cell types of the retina. And the most beautiful thing that happened in the last few years is that now we can grow the stem cells in suspension, in what we call 3-dimension, into these mini organs. So now we don't have individual cells, we have them all growing together in an environment, so in a niche, that makes them happy, makes them survive, makes them laminate, just like the back of your eye would. So it's very rewarding for us to generate all these cell types from all the diseases that we are interested in studying, because now we have thousands of these cells to grow and have enough material to test new therapies, but first of all, to really understand what's happening in the disease. Before, we only had animal models, so we had fish or mouse that we would study. And, very valuable models. We obtained a lot of information with these animal models, but with human organoids now, we can understand what the human mutations causing to the organ, right? So now we have mini human eyes basically growing in the dish. So, just a summary, why are they useful? We can, as you know, we can't get any piece of your eye or your brain or any other organs to grow in the lab. So now with the iPSCs, the stem cells and growing organoids, we can bypass that. We have a lot of tissue to grow in the lab. Or we can form the cells necessary for vision. We can grow thousands of these organoids again and study diseases, and we can then test new therapies, which is what we're doing in the lab. So to study this, as I explained, now we are generating the stem cells out of patient cells, and we have done quite a few nowadays, and we grow the organoids. So we ask the question, what is that mutation, what is the retinal dystrophy causing to the cells in the retina? So once we know that we have a defective organoid growing. So this is, for those who can see, there is a picture of a retinal organoid, and a retinal organoid, that how we see it down the microscope with the photoreceptors, so the lighting sensor cells on the outside of the organoid, and then the other cells on the inside. And let's say if this organoid has Stargardt's disease, we can see that the photoreceptor cells are starting to die because they have toxicity building up inside them. Perhaps there is high levels of oxidative stress on this cell. So we can basically now study what's happening, which we couldn't before. We would do it before in animal models, as I explained. So let's say this disease organoid with the faulty gene has dying rods and cones. We can come up now with a gene therapy, right? So that viral vector that Lauren explained, which is basically just a truck that can deliver a cargo inside the cells. And we can actually tell this truck to go to specific cells, go to photoreceptor cells only, or to other cells. So is that refined? and then the gene will be delivered inside the cells, and hopefully that gene will be on and will make protein, and that protein hopefully is functional. So now, after we replace that gene and the protein is working, we hopefully have an organoid that we can see that it now has cells that are surviving and is healthier than before. And nowadays, these type of studies is sufficient to actually take a few of these therapies to trials. In a few years ago, we wouldn't be able to do that. We would have to go all the way from lab studies, small animals, large animals, and then start clinical trials. The more and more we develop this technology and we develop the mini organs that we are growing in the dish so that they're more, they copy the organ better, we can use this as proof of concept for therapies coming in the future. So this is very exciting for us now. Another work that we do in the lab is to now get healthy cells. So now we are going to generate stem cells from healthy individuals, okay? We'll make the stem cells out of the blood, we'll make the organoids with the healthy photoreceptor cells, and what we can do is to isolate these photoreceptor cells out of the organoids and transplant them back into the eye. And that's what we've been doing. I've been doing it for the last 15 years. And we have shown that actually this, the transplantation of a purified population of photoreceptor cells, so cones and rods, can actually rescue visual perception in blind mice. And we have a poster that explains that in more detail. So please check that work that we are doing. So it's very exciting. And in this case, you need to think that it's unrelated to a gene. So anyone that has advanced degeneration or is losing their photoreceptor cells would be able to receive these cells. With gene therapy, it's one gene, one treatment, and we are developing that. There's numerous clinical trials happening, but it's literally a whole group, a whole lab is studying a specific gene that will then become a product. For stem cell, for cell therapy, it's more broad, because basically we'll replace the cells. So, as I said before, the beauty of the organoids is that we can generate millions of these cells. And what we are working on now is to actually develop, and here, I'm not using my slides that well today, but here it's a picture of the back of the eye of a mouse that has been transplanted with the human cone cells. So in green are the human cells, and we really repopulate the retina. In this case, these blind animals had no light sensing cells left, no photoreceptors left, and we were able to transplant the cells. And these were the guys that we examined, and we saw that they could now sense some light perception using behavior studies in the lab. And here it's one example of that. So what we do is it's a very simple experiment. It takes time, and you can talk to the researchers about it. I always make it sound very easily, but it's actually not. But we have an arena with a dark side, a light side, and we put the animal in there. And of course, mouse are nocturnal animals. So what they do, first thing they do, they explore the arena, but then they want to go into the dark side because that's where they feel more comfortable. We have a camera that can film and track the animals, how they move. And our study has shown, so the animal that it's blind doesn't have any preference, so it stays 50% of the time in the light, 50% of the time in the dark, and that's it. The ones that are healthy, which we call wild-type animals, they spend the majority of their time in the dark as expected. The ones that we transplanted the cells, then now are spending more time in the dark. We also had some really nice, what we call control experiments. We had some photoreceptor cells that we developed from patients with non-functional photoreceptor cells. So these cells are normal, but they don't function, so they can't sense light. So we transplanted these animals with those non-functional cells as well. and to show that actually our rescue from the normal cells was specific, and from the normal cells, we use these sham controls and were able to see that they cannot discern light and dark. So the sham treated animals now with the non-functional cells again spend 50% of the time. So we know that the treatment is related to the cells that we transplant, the rescue is related to that. So there's a lot of other experiments that we can do in the lab to assess that. So what Lauren mentioned before, we actually now combining cell therapy with electrical stimulation to try and improve this rescue of vision that we see, the promise of the cell therapy together with this new technology that is coming around. So we think there's a very exciting times for cell therapy. Another thing that we are doing in the lab is to grow, because we grow so many organoids, and believe me, growing organoids is not a trivial task. We have to grow these organoids for 7 months if we want to study disease, or 4 months for transplantation. Because these organoids actually mimic really well the development of the eye, we go all the way from stem cells to a mature, relatively mature retina. This takes 7 months. So every day these guys here give food to the cells, they need to change the media of the organoids, they need to keep them alive. And what we're doing now is investing in robotics, so we can automate the process and have the researchers actually spending more time in actually doing the fun stuff, which is analyzing the data, doing other analysis rather than just giving food to the cells. So this will also enable us to move into the clinics quicker, because these type of machines can go into rooms that are compliant to the use of, so they are good manufactured practicing rooms, so GMP rooms, where we can now grow these cells in a way that can be used in humans. So all the way that we grow now, the organoids, the stem cells, they are grown in our standard laboratory, right? So if we want to use the cells to treat patients, we need to now change everything. So we need to remove animal products, we need to go into cleaner rooms, we need to understand how we're going to cryopreserve the photoreceptors before it goes into the patient. So there's a lot of documentation, paperwork in place. So we have a dedicated team that is doing that work now to move our technology, our differentiation, our protocol to GMP. And the robot will also help on that, because a great part of our work is that manual work that I just described, and the robot can perhaps help with that. So our hope is that by moving, so it's a stepwise process, right? We still have quite a lot of basic research going on in the lab with gene therapy, cell therapy. We're slowly moving into GMP, trying to make this compatible with human use, and we hope that in the near future we'll be able to move into trials for cell therapy here in Australia. So I always like to say that this is a under construction area, the cell therapy. There are places all over the world developing this therapy. So UK, it's one of them. We started there quite a few years ago now. The Japanese are quite advanced. They actually transplanted 2 patients to date with a piece of the retinal organoid, and the results have been quite good. In America, of course, there's groups trying to develop cell therapy as well, and I'm very proud that I have my team here now developing this technology in Australia, because as I always say, I want you guys to have access to it, and I know how hard it would be for you to gain access to any clinical trials all over the world, so I really want to make it happen here for you guys. And with that, we also have Vision Loss Priority Setting Partnership. So the same research, the same enterprise that we are building to get this work into clinical trials is also interested in hearing from you. We cannot do the research without you. So we have this study, and Eden is leading it with Kate, and she has a poster, go there as well and understand. But what we're doing is to trying to identify the top 10 research priorities. What is it that you guys want us to study, right? Please let us know, because this is what we're going to do for you guys in the future. And that's it. Thank you very much.